You're watching Mirror 7 News. Stay informed, stay ahead for more news.
Let's talk about something that could reshape how we understand the origins and risks of future coronavirus outbreaks. Bats. Specifically, new research published in Nature Communications has just highlighted a striking pattern in bat populations. Immature bats, like juveniles and subadults, are shedding multiple types of coronaviruses at the same time and in sync with one another seasonally. This study took place over a span of 3 years in eastern Australia, focusing on flying foxes, mainly black flying foxes, that is BFF, and some gray-headed flying foxes, that is GHFF. By collecting over 2,500 fecal samples from both individual bats and under roost sheet samplings across 5 locations, scientists were able to identify 6 different beta coronavirus clades. All of these belong to a subgroup called nobecoviruses. What's especially intriguing is that 3 of these clades seem completely novel. They've never been found before. Now here's the big deal. Co-infections, or infections with more than one virus at a time, were found to be most common in younger bats, especially around the time they're weaning, and not just a few cases. The study points out that these co-infections were not only frequent, but also happen seasonally and in sync across multiple viral clades. Why does this matter? Because when different coronaviruses infect the same host at the same time, there's a much higher chance of viral recombination. That's where pieces of different viral genomes mix, potentially creating a new strain. It's like a viral genetic remix. And this is often how new variants with zoonotic potential, that is, the ability to infect humans, are born. One particularly telling case involved a juvenile bat infected with a recombinant virus, mostly one type, but with a spike protein section from another. That kind of recombination could be how future human-targeting viruses evolve. The data also revealed that these viruses circulate throughout the year, but spike between March and July, coinciding with early autumn to midwinter in Australia. What makes this study so robust is its combination of 2 sampling techniques: under roost pooling, which is efficient for wide surveillance, and individual bat sampling, which gives detailed, host-specific data. Together, they provided a much clearer view of how the viruses behave across time, location, species, and age. Interestingly, one clade, 2D.3, appeared almost exclusively in GHFFs, suggesting specific host-virus relationships that can guide us in future surveillance. So what are we learning from this? First, bats are critical reservoirs for coronaviruses, and their viral shedding isn't random. It follows seasonal, age-related, and species-specific patterns. Second, immature bats are hotspots for viral mixing. And third, understanding these dynamics might help us predict, or even prevent, the next coronavirus spillover into humans. In short, if we want to stop future pandemics at the source, studies like this are our early warning systems. They're showing us where to look, when to look, and what to watch out for. Stay subscribed for more videos.